Hi everyone, welcome back to class. Today we are going to finish up talking about acetylcholine by covering the systems and receptors involved. So first, just kind of a review of what systems acetylcholine works in. So we talked a lot about that neuromuscular junction where we get the cholinergic neurons. Of course, the central nervous system, it plays a big role. But another place that we haven't talked about yet is the autonomic nervous system, or the ANS. So in the autonomic nervous system, then the preganglionic neurons of both the sympathetic and parasympathetic branches are cholinergic, and the ganglionic neurons of the parasympathetic branch are also cholinergic. So, here's, again, an overview. You should be familiar with the sympathetic and parasympathetic nervous systems. On this side, we have the sympathetic division. Over here is the parasympathetic. Now, the preganglionic neurons, so those are the neurons going from the spinal cord to the ganglionic cells. So, kind of, this little area here, that's that preganglionic. And in both the sympathetic and parasympathetic, they are cholinergic. So we see that with blue. In the parasympathetic nervous system, so that's that conservation and relaxation stage, then the postganglionic neurons are also cholinergic. So that's represented by these purple lines. Okay. We will talk about the postganglionic neurons later on when we start talking about noradrenaline, which is involved in that fight or flight. And it'll make a lot of sense just based on what we know about adrenaline or whatever we're going to call it. Okay. But for acetylcholine, it's that calm down, that processing things, conservation, relaxation. Okay, so we'll see the pupils will constrict, decreases that heart rate, constricts airways, increases salivation, stimulates digestion, and it helps store glucose. Okay, so all of that is involved in that parasympathetic division, and all of that is aided by those cholinergic neurons. So again, here, just the same thing, central nervous system to the ganglionic neurons, that's all going to be cholinergic. So those preganglionic neurons will release acetylcholine. However, in the parasympathetic nervous system, then the ganglionic neuron will also release acetylcholine onto that target organ. Whereas in the sympathetic branch, the ganglionic neuron will release norepinephrine. You'll notice that I've kind of switched between norepinephrine and noradrenaline. In this class, we're going to call them the same thing. Just familiarize yourself with that. When we get to that section, I'll also cover that for this class, we're going to noradrenaline, norepinephrine are the same thing. Okay, so what about some specific agonists and antagonists of our cholinergic neurons? So we have different type of receptors that cholinergic neurons act on. One type of receptors they act on is the muscarinic receptors. Okay, so agonists of these muscarinic receptors, which if you don't know what they are, we will cover in a little bit. But they are parasympathomimetic. I always struggle with that. But pretty much an agonist of the muscarinic receptor mimics the effects of the parasympathetic nervous system. Okay, so an agonist of the muscarinic receptor will calm and digest, whereas an antagonist will inhibit that parasympathetic function. Okay, so it's going to act more like a sympathetic nervous system. Okay, so it's going to be more fight or flight. It's going to prevent that rest and digest. Okay. Again, this should make sense just based on an agonist would mimic, obviously, acetylcholine. So if acetylcholine is involved in parasympathetic, it would do parasympathetic things. If you block it, it's going to prevent parasympathetic things. Okay, so here is a brain. This is a rat brain, I believe, some kind of rodent. And what it's showing is multiple different areas involved that have cholinergic neurons. Okay, there's a lot going on. We're going to walk through it. But a lot of it is broken down into kind of three big things. So here we've got that BFCS. That's the basal forebrain cholinergic system. This is a very large system involved in the cholinergic neurons. Okay, know this. You should understand, recognize BFCS. It projects from the nucleus basalis, the substantia insominata, the medial septum, diagonal band nuclei, to the cortex, hippocampus, and limbic structures. Okay? So, I read off all those, and you can see where it goes. Wow. Wow. How exciting. This is involved in learning, memory, and attention. Okay? This makes sense when, remember, we were talking about dietary choline and how not enough dietary choline may lead to deficits in these three things, how certain disorders such as Alzheimer's show deficits in choline and deficits in these processes. Oh my God, it all connects. So that's the BFCS, big one. We also have the striatum. Okay, the striatum is involved in motor function. Think of the striatum, we've got the motor cortexes. Really, that's it. So that just makes sense. Now, interneurons within the striatum, specifically the caudate putamen, are affected in Parkinson's, right? So balance activity with dopaminergic systems that degrade with the disease course. However, in the early stages of Parkinson's, then there actually seems to be the balance between acetylcholine and dopamine. So we'll talk about Parkinson's again when we talk about dopamine, but it is interesting to note that it's not just dopamine involved in Parkinson's. It's the balance between acetylcholine and dopamine. So you need both of them, okay? So that's what this was pointing out right here. That's the caudate putamen, okay? As well as the nucleus accumbens. And then we've got our dorsolateral pons, okay? So the dorsolateral pons, that's these green areas here, those are involved in arousal and addictions, okay? Now remember, arousal doesn't mean like sexual arousal. It could, but it's types of arousal, okay? So it could be like excitement, it could be fear. Those are all types of arousal, okay? So this is the reticular activating system. It's involved in arousal and addictions. So the dorsolateral pons has cholinergic neurons within it that help regulate arousal and addiction. So you will need to know these three big areas, okay? And what systems are they involved in? Now, let's kind of break some of those down so that we know more about them. Again, the big one is the basal forebrain cholinergic system. Okay, so these cholinergic neurons are located in that medial septum, in the diagonal band of the nuclei, in the nucleus basalus, and in that substantia innominate. I really should have taken Latin, but I didn't. These project to big areas like the cortex, hippocampus, and amygdala, okay? So again, cortex, learning, hippocampus, memory, amygdala, emotion. Makes sense what they're involved in. We see that loss of these cholinergic neurons in this system is associated with cognitive decline in Alzheimer's disease. So, memory and attention are implicated in this system. Again, cortex, amygdala, hippocampus, those areas are involved in memory and attention. Just makes sense. Okay? Now, what if we want to mess around with it? Okay, let's add some drugs because that's what this class is about. So we're going to start by talking about atropine. Okay, atropine. I don't know. It's a plant alkaloid that's found in nightshade. Okay, so this is nightshade, or Atropa belladonna. And it is an antagonist of the muscarinic acetylcholine receptor. So because it's an antagonist, it's going to prevent the parasympathetic system, right? So instead of constricting the pupils, it dilates the pupils. Okay? It does the opposite pretty much, or it prevents the constriction of the pupils. Okay? It is able to pass through that blood-brain barrier. So that is why it can be deadly if you take too much of it. Now, oral administration tends to inhibit cognition in animals. Okay? If you give it to an animal and then try to teach them something, they can't learn. Now, because I love history, a little fun history of it. These history things probably not going to be on the exams, okay? Just know, like, how they work, but I like the history. So, the name Atropa, right here, is from Atropos, which is the goddess of fate, responsible for method and timing of death. Yeah. Belladonna is Italian for beautiful woman. And that name's been used in a lot of cosmetics. And actually, historically, women used to take nightshade, like purposely eat it, for cosmetic use in order to dilate their pupils, because that was seen as a beautiful thing. You know, those dilated pupils. Oh, beautiful. Love those big eyes. So, people used to actually take nightshade for those properties. Obviously dangerous, and we'll talk about a lot of these drugs or toxins throughout the course that were used for cosmetic purposes, and now we realize that was a bad idea. Especially because with long-term use of nightshade, it can actually lead to visual distortions or blindness. So, don't recommend. Okay. What's the role of acetylcholine in the BFCS with cognition? I mentioned that we could administer atropine or nightshade. Atropine is the toxin of nightshade. Or related drugs like scopolamine, which are amnesics in humans. Okay, so produce amnesia. So scopolamine and morphine was a drug combination that used to be administered during labor. Pretty much it caused women to kind of forget the pain of labor. So they'd still experience the pain, but then after it was over, they'd kind of almost wake up with their new babies and forget the process of labor. So they don't have any memory of it, and then they're more likely to have more kids and do labor again. So acetylcholine is then thought to be important for that memory consolidation, especially episodic memory. Okay, and it's thought to be kind of focusing on the hippocampus, which remember is one of the targets of the BFCF. Alternatively, it's been proposed to affect memory through effects on attention and sensory discrimination. So rather focusing on that hippocampus, more on the cortex, the frontal lobe, and really causing you to just not pay attention to stuff around you. So like during labor, you're not paying attention to the pain. You're not paying attention to the labor. So because you're not paying attention to it, your brain's kind of like, well, it's not important then, so there's no point in, putting it into long-term memory. Obviously, we no longer use scopolamine for labor. Okay. Now, there have been some cool research studies using animals to look at the effects and the role of the BFCS in cognition. So one is using microdialysis. Then research showed that our acetylcholine levels actually increase during tasks that require sustained attention, more so than in tasks that require only like partial attention or like short-term attention. Okay. So this was a operant control task. So they get rewarded for the task. So like press the lever, you get food, that kind of thing. And using different schedules of reinforcement, then we're able to adjust and see how well these animals do. Okay, so there's an operant control task, so press the lever, get food. There's an F19S schedule of reinforcement, so this is a fixed interval 9 seconds. Okay, that's what that stands for. Operant reward task, that's a predictable delay. And then the sustained attention task. Okay, so these are 3 different types of tasks. And what you can see is that all of them, on the x-axis, we have baseline, the B's, and the task, so would be the T's, obviously, and then acetylcholine amount, so the percent change from baseline. So around baseline, then our 3 groups, have the same amount of acetylcholine, because obviously it's baseline. But during sustained attention tasks, then that amount of acetylcholine increases drastically, whereas with our 2 different operant control type tasks, then we still get an increase in acetylcholine, but it's not as drastic. Okay, so this is suggesting that acetylcholine is specifically important for the sustained attention. Not necessarily that, this isn't proof that it's important for memory consolidation, but we're showing its importance in attention. Okay? So, now we'll continue. Up next, we've got saporin. So saporin is a toxin that's been isolated from the soapwort plant. And it is a water-soluble toxin, so it's able to move throughout your body pretty easily. It is highly specific inhibitor of ribosomal function. Okay, so it gets into that cell, blocks function of those ribosomes, specifically ribosome inactivating protein. And it can be conjugated to antibodies to target specific cell types. Okay, so we actually will use it to investigate anti-cancer agents. Now, this is, you probably haven't heard of saporin much, but you've probably heard of its cousin. It has a very popular cousin called ricin. Okay, so ricin is a protein isolated from the castor bean, and it is a related ribosome inactivating protein. Okay, so it's another type of RIP. And it is a poison used especially during the Cold War and even after. Great example in 1978. Then a Bulgarian defector, Georgy Markov, was assassinated in the streets of London by agents of the secret police of Bulgaria. And he was actually working with, the secret policeman was working in concert with the KGB during the Cold War. What? They actually modified an umbrella to fire a tiny pellet into this dude's leg that was covered in ricin. And because ricin is untraceable once it enters the system, then Georgy Markov didn't know what was happening, and then he just died. Because it entered the system. It's scary. Again, ricin, more popular or more famous than sporin. But both sporin and ricin work in cholinergic cells. Okay? So, you're probably like, none of this has to do with it. But let me explain a little bit more. So, this is a specific type of sporin, 192 IgG sporin. Okay, you don't need to know the specific, just know sporin. But it's a conjugated antibody that targets basal forebrain cholinergic neurons. Okay, so sporin, I mentioned here, is a RIP. Okay, but we can use it to target specific cell types. So we don't use sporin or ricin or whatever for their like poison stuff. We use it as a targeting system for research purposes only. Okay? So we administer this specific sporin strain, and what it does is it targets the BFCS neurons and then selectively lesions only those cholinergic cells. Okay, so this is a specific type of sporin that we have altered that only targets the cholinergic cells of the BFCS. And what that does is it produces specific deficits of learning, memory, and attention. Okay, and this is how we were able to kind of highly support, highly conclude the role of BFCS in learning, memory, and attention. Because when you take it away, right, you take away the BFCS, you lose or you get deficits in these three areas. Okay. So what this diagram is showing is a Morse water maze in rats. So the day is on the x-axis, the y-axis is the path length. Over time, control rats, you can see here in the purple, their path length decreases. That's what we expect. But in rats that were injected with this sporin, okay, when they were injected in the nucleus basialis, which is one of the areas of the BFCS, you can see the path length. It did decrease, but it took much longer, right? But specifically when we inject it into the cerebral ventricles, then the path length takes much longer to decrease. So it's inhibiting the learning and memory, okay? Especially that spatial memory. It's really focusing on. In sustained attention tests, in animals that were lesioned with sporin, so we talked about sustained attention tests before, then we also see that these animals treated with sporin had a lower number of correct hits when the signal strength was weak, suggesting deficits in attention. So these animals weren't able to have that sustained attention as much as the animals that didn't receive sporin. Okay, so performance was comparable on other aspects of the task. So there doesn't seem to be a global cognitive impairment or global motor impairment. The animals can still walk, whatever, fine. They can still, think, do normal cognition, but they struggle with attention. Okay, so I'm going to end this video here. I know it was a little long, but we'll end this video here, and on the next one, we will continue finishing up some of those acetylcholine systems and then start talking about the specific receptors, which I know I've kind of hinted at.